Welcome to the Cell Cycle series. This video focuses on cancer. At the end of this video, students should be able to describe what cancer is and how it affects the cell cycle. Cancer is a disease affecting a cell's ability to control cellular division. Around 10% of cancers are hereditary, which means that an individual has a genetic predisposition to getting a mutation that someone in their family experienced. For example, in a family where women experience breast cancer, a gene can be passed down from parents to offspring. This gene is known to mutate more often within individuals that carry it. For example, the BRCA1 and BRCA2 genes are known genes that can cause mutations that lead to cancer in the breast. This mutation in the gene can lead to breast cancer, but does not guarantee that the individual will get the gene, or that if they do get the gene, that they will get cancer. All it means is that they have a genetic predisposition to getting the gene. And if they have the gene, they have a genetic predisposition to having a higher chance of that gene mutating, leading to breast cancer. This predisposition is just a higher chance of the mutation rate over someone who doesn't have the gene or who does not have the mutation in their genetic line. Other forms of cancers are acquired by means other than heredity. About 80% of all cancers come from exposure to carcinogens. Carcinogens can be chemicals and toxins, things like UV light. These are things that mutate somatic cell DNA, which is after conception. It hijacks the cell's control mechanisms, and the cell can no longer control cell division. Cancer is not only a human disease, it also plagues the rest of the animal kingdom. Cancers start very small with one or very few mutated cells undergoing divisions and passing the mutations on to their daughter cells. Over time, this series of divisions begins forming a benign growth. This benign growth is not cancerous and can eventually become a tumor. A tumor is simply a grouping of cells that is experiencing uncontrolled cell growth. A benign tumor stays in one type of tissue in one area of the body. Often it's encapsulated. Some tumors can become malignant or cancerous. They lose control of the ability to regulate cell division and also gain the ability to invade neighboring tissues. In most cancerous cells, the shape and the size of the cell are different from the normal version of the cell that they mutated from. As the cancer cell proliferates, they eventually perforate the tissues that they are in and branch into the neighboring tissues. Finally, these cancerous tumors can metastasize and spread throughout the body. As the cancer cell continue to proliferate through the neighboring tissues, some pieces of cancer cells can flake off and cross into the blood, the lymph system, and the interstitial fluid. These can carry these cells throughout the body and can cause cancerous tumor growths in other areas of the body distant from the original tumor. Proto-Oncogenes regulate gene expression. When there's a mutation in a proto-oncogene, it cannot turn off the gene's expression and uncontrolled cell growth occurs. These mutated forms of the gene are called oncogenes. The mutations that result in oncogenes can be simple missense mutations, which is a one base change, substitution, addition, that leads to a different amino acid being coded for. Although translocations of sections of a gene where that section enters a different gene can also be a culprit that leads to those types of cancer. Tumor-suppressing genes can also malfunction, creating cancerous tumors. Typically, the suppressing agent will check for mutations throughout the genome, repairing any malfunctions to maintain the genetic integrity. Checkpoint proteins like p53, which is sometimes referred to as the guardian angel gene, can be tumor-suppressing genes, which arrest the cell cycle when mutations are spotted. This can lead to proteins fixing the mutations or to apoptosis. 50% of cancer types come from mutations in the tumor suppressor genes. The RB protein that regulates E2F acts as a negative regulator for transcription of a gene. When mutations arise, E2F is always active, and constant transcription and subsequent cell division will occur unregulated. Most often, cancers arise from a series of 10 or more mutations in the same cell line. Usually, this occurs through repeated exposure to carcinogens, often the same carcinogen, like in smoking or UV light. Often then, cancers are found in older people because those mutations take time to accumulate. Malignancies can also accumulate mutations that can lead to additional changes in the cancers. Students should be able to differentiate between mutation, benign tumor, cancerous tumor, and malignancy. Students should be able to describe how cancer affects the cell cycle. Students should understand negative control and tumor suppressor mutations. And students should be able to indicate the difference between proto-oncogenes and oncogenes.